We're beginning, and we have begun. Hello, hello. All right, so we're picking up from where we left off, and where we left off, just to pop over there, was these historical figures, and this is a really interesting area to talk about, and you can see the notes are there, so you don't have to. You can listen as well. So we talked about Jenner and the important discovery of smallpox prior to Pasteur, when they were part of the effort, and this one is very, very telling, was a physician named Ignaz Semmelweis, who was in Vienna in Austria. For those, history's a little weak. In the 1850s, Vienna was everything. Vienna was art, history, culture, science. That's where Mozart played. That's where Beethoven went to play. I mean, if you were a leader there, the main hospital, Semmelweis was the lead physician, and he made an again scientific method, made an observation. He observed that they were looking at something called puerperal sepsis. That means childbirth fever. You'll learn that at the time it was principally a streptococcal infection. And he observed that when midwives were delivering, they had a very low rate of it. And when doctors and medical students went up to deliver babies, they had a much higher rate. And he observed that the medical students and the doctors were coming up from the laboratory. Well, it's not like the micro lab. The laboratory meant they were cutting up dead bodies. Okay. And they'd have a rubber, if you had gloves on, they were lucky. They'd have a rubber apron on. That was about it. And they were covered in yuck. And they're going out there doing vaginal deliveries. So you can imagine how close the contact would have been and easy to spread that, because he knew a friend who had died of a streptococcal infection and saw similar things. Didn't really know it was streptococcus, but saw similarities in the infection. So that was sort of the backstory behind all of this. So at that point, he said, well, since I'm the boss, I'm going to have medical students and doctors not do that. They're going to have to wash their hands. We're going to put some, basins of water and different soaps and have them take all that stuff. And it improved dramatically. Again, he didn't know why. He just suspected they were passing on some type of infective property. And he wrote a paper on it. And the paper was, basically saying that because we were doing this, we need to take more steps to be clean, etc. You would think he'd be celebrated. Well, he broke the cardinal sin of doctors. He said, well, we are causing the problem. And they ostracized him, just to show you. Even though he helped women survive and babies survive, and unfortunately, he tragically died years later of, interestingly, an infection. Go figure. So it's kind of an important story with a very unhappy ending for him. And that was before that. It was the midwives versus the doctors. So it really started to underscore that. And around the same time, with Pasteur, Lister, who was inspired by Pasteur with his experiments, basically was the father of what we call modern aseptic technique. And this is my spin. When you hear the word sterile technique, sterile is pretty hard to achieve unless you're in an operating room. And the operating room isn't by any means sterile. The equipment is, and the gown, and the gloves. And you'll learn about it. But really, the concept is aseptic. When you wash your hands for surgery, you try to wash your hands to get them clean enough that you can pick up the inside portion of a glove that's going to go against your skin, the sterile portion of a glove that's designed not to go on the patient, but on the outside. I mean, there's a whole lot to it. Nevertheless, so what Lister saw what they were doing. He saw that they basically, maybe if you were lucky, they wiped off the instruments. They didn't clean off the patient. They didn't wash them off with anything resembling anything that we would call an antiseptic or a disinfectant. They didn't wash their hands, the instruments, the patient, etc. when they did surgery. So what he did was, inspired by Pasteur, who we're going to get to momentarily, basically said, listen, we need to start. So he used dilutions. It happens all the time. He used dilutions of what was called carbolic acid, known as phenol. And for the longest time, we still to some extent use phenol products, though not nearly as much. Phenol is an interesting compound from organic chemistry that when you dilute it, enough, it becomes a really good antiseptic. The trouble is there's a certain amount of absorbance, and at higher concentrations, it's pretty nasty. You get a pretty nasty, it's called a base burn. It looks like an acid, but it's really something called the conjugate base for you fans of chemistry. But long story short, what happens with that was that by using these diluted phenol solutions, he was really able to achieve a tremendous amount. And most of what you'll do for many of you in a hospital or in a medical environment or in an office or anything that you do, you'll learn a lot about using that. And I mean, they used it in a product that bears his name, Listerine, which is an herb mouthwash. And for the longest time, phenolic compounds were the active ingredients in antibacterial soaps. It's changed. We'll go over all of that when we do that unit on antiseptics and disinfectants. So, because again, there's a certain amount of absorption that they've discovered over time that can be problematic. Which brings us to Pasteur, and he was the definitive thing. Okay, he was confident because he, in the early days, he really used the microscope a lot. So he took little pieces of material and looked at them under the microscope and samples of things to see what was inside there. So he was really an advocate of the microscope, and he had credibility. The reason he was so well known, even though he was a physician, he had helped the wine industry. The wine industry in France was floundering, and the wine was spoiled. And what to use basically in the fermentation process of grapes, sugars into alcohol. Pretty standard fermentation, stuff you're going to do in the lab all the time. And what happened was the fermentation process kept going on even after they bottled the wine. And if they didn't drink the wine rapidly enough, it got sour because it goes from alcohol, and the longer that the fermentation goes on, it becomes acidic. And the acidic tastes sour. That's what acids do. And so he was trying to figure out, and he said, if we try to heat the wine and we bring it up to boiling, it doesn't taste good at all. It tastes rotten. And he started to experiment with different times and different temperatures in an effort to, because he saw, when he looked at the wine, he saw little yeast particles under the microscope. He said, I've got to find a way to kill these yeast particles without clobbering the wine. And he basically experimented, and he roughly, at about 140 degrees Fahrenheit, when he held it for about 30 minutes, not boiling by any stretch, boiling is 212, and if you do and basically it's approximately 62.5 degrees Celsius. Without doing anything in science, you've got to do Celsius. Make sure. It's basically the same thing, and he basically, it didn't lose any of its flavor or the inherent acidity content, but gave wine what wine is supposed to be. And basically, he saved the wine industry, and they coined a term that we still use today: pasteurization. So he already had a lot of credibility. If you're, just to give you an idea how important that is, it's not a form of sterilization. We're going to go over this again and talk about the way we vary it today. But it's basically what your dishwasher does. Your dishwasher pasteurizes your dishes. Your dishes aren't sterile. Internal temperature of the dishwasher is approximately 140 degrees Fahrenheit, and it holds that temperature at least for 30 minutes, if you did not know. So you can understand the importance of that process. And basically what it does is it destroys typical bacteria and organisms and entities that can cause basically foodborne illness. For them, not all of them, but most of them, just to give you an idea. So already he was important. So he's trying to find a way to convince people what this was. And the big deal was about the air. You might remember I talked about Reddy with it. Well, you had the fine mesh gauze or the cheesecloth. Well, but we can't duplicate it. And, so there's some kind of life force in the air. So I asked to describe something to prove how something can stay quote unquote sterile without while it's exposed to the air by some type of method. And this is why I cooked up this other PowerPoint, because it's got a better illustration. Here's the illustration. He basically had the glass blowers make a flask. He experimented with different flasks. And he experimented by putting little pieces of cotton in here. But with the cotton in there, it would theoretically might block the air, even though it didn't, but that's people would say. And when he was able to trap particles, when he heated the material, the liquid that was in there in an effort to sterilize it, and he would sample the cotton to see what was trapped in there. And he was very confident, based on looking at the microorganisms, that basically as he was heating it, they were being suspended and being trapped in the cotton. So he had to try to figure out a way to do this. So he took a flask, and what they used were called broths. This is very relevant to what you're going to do in the laboratory. How do you know when you get a tube of, quote, broth or liquid in the lab, how do you know it's sterile? It's clear. You can see through it. If it gets cloudy, it means there's stuff in there and the light can't get through. It gets kind of murky. Okay, we call it turbidity. You can even measure the turbidity index in the lab with a spectrometer. We typically don't do that with our lab experiments unless Dr. Dress has changed. Did you like Dr. Dress? You haven't met you today? Yeah, it's great. So you have a good time meeting him. Nevertheless, so they worked with broth, and the idea is he put a broth in there, boiled it to sterilize it, and it stayed clear unless he tipped or broke it. And you see the way they had the neck, they made it so it was open to the air. So the air could not only escape, but that swan neck, he theorized that it would trap the particles there, and as long as they didn't come in contact with the broth. And he was absolutely right. Except you couldn't duplicate it. He made two significant errors. The broth that he used was a combination of yeasts and sugars, very similar to what he was working with pasteurization in the wine industry. So when other experts, other scientists began to do this, they couldn't remember, to duplicate something. So Tyndall and Cohn, one was English, one was German, experimented and couldn't duplicate the results because they used different broths. The most common broth to use was something called a hay. Okay? They were used a lot. So infusions, you're making a coffee or tea, effectively it's an infusion. You're using warm water and you're extracting flavor and elements from whether it's tea leaves or coffee grounds, whatever. It's all really is. So hay infusions were something that they worked with a lot for whatever reason. And they take hay after it was made and they pick it up off the ground, the soil, and they put it in water and let it heat it and let it steep, and voila, there would be a hay infusion. And whatever they used, if they used his broth, which was yeast and sugars, it always worked. If they used hay infusions, it didn't work. So there was a lot of controversies. And then Tyndall and Cohn started to look microscopically, and they identified particles in the hay infusion, and they named them endos. So that was the first element that Pasteur didn't have right, but his premise was good, was he couldn't duplicate it because he was using a very specific broth that was very amenable to sterilization, because you had just yeast and sugars, which are prepared, yeast are prepared, yeast kill, hot water kills yeast. Have you ever baked or made pizza dough? When you want to activate the yeast, if the water's boiling hot, they're dead. So those endospores were the problem. The other problem was that boiling doesn't kill things. It doesn't kill everything. It's only in the movies they say, oh my God, she's having a baby, go boil some water. I mean, it's useful. It's better than not having anything, but boiling is not a form of sterilization. Never is, never was. And the reason is spores. Okay, so endospores are everywhere. So endospores were involved. And indeed, when we get to Koch, who was somewhat of a competitive rival, though younger than Pasteur, he came after Pasteur, he was the lab geek. Pasteur was much more of the how can I help people kind of a guy. He coined the term, as I'm probably told you before, virus. He identified the first treatment for a heart for potentially for uh, different kinds of viral diseases that were present. He was really early on involved in those types of things. So he started to look for when people were infected, particularly with rabies, which is a viral infection. He actually started extracting serum from animals, a component of the liquid portion of the blood for animals who had survived rabies bites and were injecting patients with it somewhat successfully. So he actually was dealing with something called antitoxins at that particular point. But long story short, he was able to find a lot of different things for treatment. So that's why the Pasteur labs were sort of pioneers in varying kinds of immunizations and different kinds of treatments. So herein is the problem, the relevance of endospores today. Endospores are effectively, and you'll learn about this in the next chapter, which is the chapter sort of on the anatomy of bacteria. A limited amount of bacteria have endospores. Endospores are effectively indestructible. They're everywhere. They're on this carpet. They're on the bottom of your shoes. They're in the soil everywhere outside. That's why they're in the hay infusions. And they have, as you can see, these are the names. Family name of most of the endospores, the ones that cause the problems vis-a-vis Pasteur in the soil, were Clostridium tetani, Clostridium botulinum, botulism, Clostridium perfringens, the S gangrene when it gets into an open wound, and the one that you folks, if you're in nursing in the hospital, are going to deal with the most, Clostridium difficile, named because it's such a pain in the neck to get rid of, affectionately called C. diff. See, when you have with nomenclature in bacteria, Staphylococcus, capitalize that, aureus, small letter, and aureus comes from because it has a golden color of a pus. We can call that S. aureus. So it's fair game. You don't have to do the first name, the genus. You just need to have it capitalized, the letters, and it's fair game on an exam, by the way. So, or a medical chart. S. aureus is everybody knows Staphylococcus aureus. You don't even have to specify what the S is because, like, Streptococcus viridans is S. viridans. So the S's aren't necessarily the same. It just has to do with conventionality of binomial nomenclature, how to chart wise. So this was the problem. You can boil that stuff till the liquid disappears. You can't get rid of the endospores. They go into the base endospores. They have a very elaborate protein coat, and this was the problem. That's the reason why if you have a wound, and go to the emergency room or an urgent care place, they're going to ask you the first two questions. The first question is very simple. What's your medical? You want to get paid. The second question is what? When was your last tetanus shot? And most of the time, most wounds, as you will come to learn, are not tetanus-prone wounds. They are tetanus-prone wounds are ones where we have really nasty injuries with a lot of mess in them. The typical wound that you put stitches in an emergency room is a clean cut wound. And boom, kitchen knife. Get a cut, it's a couple of stitches. No big deal. Comparatively, the reason they're asking you about the tetanus shot is when you're in a terrible accident and it's off the road and nobody sees you for a while, tetanus can start to flourish right away. So it's for the next time to make sure that your tetanus current. If you did not know, you're getting 3 immunizations at the same time. You're getting a tetanus booster, a diphtheria booster, and a pertussis or whooping cough booster. All the same. Trivalent. So the first thing, and tetanus is everywhere. So any wound potentially could be a tetanus-prone wound, but the interesting thing about this particular family, the Clostridium family, is they are anaerobes. Anaerobes means they grow without oxygen. So the only time we can see those are in an environment where there has been dead tissue because that doesn't have any oxygen. So that's what I mean by it's a severe wound with dead tissue, skin that's turning black, that kind of a thing. So even in a setting when you have a clean wound, even in a surgical wound, you don't worry about tetanus unless it starts to get infected and the edges start to turn black, and there's been some really significant postoperative problem. Clostridium perfringens was something that troubled people. We don't see it as much today because we have much more efficient health care. It was the scourge of early wars. They would die of something called gas gangrene. We'll talk about gangrene. The gangrene you see the most of is not infectious in origin. That's typically gangrenous toes and fingers and legs and things like that in diabetics. Okay, that is not due to an infection. That is circulatory death because of lack of circulation. However, war injuries, exploding projectiles, exposed bone, torn apart skin, and thing that was you in World War, losing limbs in the Civil War, in the wars that intervened in World War One, even World War Two. That was an enormous problem because it wasn't until the Korean War that they started to have chopper evac and mobile hospitals. In the old days in wartime, the best you could do to try to get whoever was wounded, you had a couple of rifles and a poncho and improvise a stretcher and tried to carry that person as quickly as possible to some type of facility, some type of improvised hospital to get treatment. So there were a lot of lost limbs and things like that. We'll get into the history of antibiotics. The first antibacterial used in wartime, very famous, is called sulfanilamide. And we'll get into that when we talk about antibiotics. That's something that we, the Allied forces, had, where the Axis forces in World War II, Nazi Germany and Japan, did not have. And every soldier would have a wound pack, and they'd have a little vial, a packet of some type of green powder they'd sprinkle on the wound. It was very effective against preventing Clostridium perfringens or gas gangrene. And that had a lot to do with winning that war. Okay, and we'll get into penicillin, also had a lot to do with winning. Because the wars are typically wars of attrition. I'm a World War II buff. It's just like my favorite place to go is New Orleans to the World War II Museum. The food's pretty good there too. So if you get a chance to go, it's in what's called the Warehouse District, which is between the Garden District, which is my favorite place, and the French Quarter, which is not. The food's really good. Go there, because I could spend days there. Nevertheless, that's the saga of Clostridia. The one we worry about in food poisoning is botulism. Food poisoning is typically not fatal. Botulism is about the only immediately fatal part of food poisoning. It's called food intoxication. You're not ingesting the Clostridium, you're ingesting the toxins. And that's a neurotoxin, just the tetanus is very, very quickly. So, and it's rare. That's why we do something called commercial sterilization for certain products that are, again, needed to do that. Botulism is something we see very rarely today. The people who have to worry about it is typically the most severe outbreak that I can recall in my lifetime happened in vichyssoise, which is, by the way, cold potato soup, if you did not know. And they were making, it's a French name, but they were making it in Newark, New Jersey, right by the old Newark airport. And it was marketed as Bon Vivant vichyssoise. And they had poor quality control, and about a dozen people died who having ingested, because you don't heat the soup whatsoever, so there was no chance to kill. The toxins like that get killed pretty well by eating. But it was designed to open the can and boom. It's kind of why for a long time, and even I still do it, the can's dented, you'd be very careful. You're going to get that last can of cream and mushroom soup to make those green beans for Thanksgiving, and you're going Wednesday evening, you get a dented can. You don't want to get that can. Make it yourself. Just get some mushrooms, a little bechamel. You can do it yourself very easily. Nutmeg, perfect. You're making a bechamel. You got to put a little nutmeg in. Flour, fat, milk, happy dance.
Have you made it?
Absolutely. Who wouldn't?
Is it pretty good?
Bechamel is wonderful.
Better than the can?
Yes, always better than you can. There aren't a whole lot of things that you don't make yourself that aren't better. Though the Rayo's marinara is pretty darn. But I digress. Okay, so those are just some of the examples. And C. diff, why? So botulism is anaerobic. Where we see botulism is a lot with home canners. So if you don't know what you're doing and you're canning stuff at home, be careful. It's okay if you're doing tomatoes because they're very acidic and it doesn't grow very well in acid concentrations. But if you're, canning, vegetables that aren't acidic in nature, like a tomato would be as a fruit, you got to be very, very careful. The other thing is people who do home brew. Where we see botulism is in honey, and that's why we don't give honey to very, very young babies. I mean, I would be hesitant under, particularly if it's like unprocessed honey that you're buying at a flea market or a country store. I would wait till they're at least age 2. I've seen one of my students years ago was doing home brew with honey, they effectively making mead. Okay, and we looked at it and he goes, "What's this stuff growing?" And we stained it. Botulism. Yikes. Okay, because I mean it's deep because there's no air is getting through the honey. And the last one is C. diff. Why does that grow anaerobically? It grows anaerobically because it grows inside your colon. And your colon, for those of you who are going to have AP2 this year, is an anaerobic environment, in oxygen. And the other endospore that's really relevant is anthrax. You will work now. Anthrax is the one that we get to Koch. Basically, he utilized this and eventually won a Nobel Prize. The only reason Pasteur didn't win the Nobel Prize is he died before the Nobel Prize started in 1896. Koch won one in the early 1900s. I mean, his research and his contribution for sure. But Koch was a lab guy, and all of the techniques, not all, but most of the techniques you're using in the lab, and we'll do this are basically things that Koch developed. Use of agar gels that we use in agriculture. I use it for isolate strep, staph, E. coli, tuberculosis, and particularly anthrax, which is an endospore. Except it's aerobic, so you're going to use a cousin called Bacillus subtilis, and we call it B sub for short. And Bacillus subtilis is called Bacillus anthracis, but B sub is something we use a lot, and indeed one of the good parts about B sub, well, it doesn't hurt you, and it also makes an antibiotic called bacitracin, which is part of the original triple antibiotic, and you can buy independently. Triple antibiotic was developed in Pittsburgh by a company called Burroughs Wellcome with neosporin, which was his name, would contain neomycin, bacitracin, and another antibiotic called polymyxin B. Historically, it's changed a little bit. When we do the antibiotics, I'll talk about that as well. Okay, so he basically, his lab solved a big problem that was happening in the agrarian populations of Europe. So around the time he made this discovery, let's say around 1900, okay, it was a farming culture. There was a lot of industry, certainly, but anthrax was a disease that was commonly spread cows, sheep, goats, easily spread. And the only time people were getting it is if they were basically they were shearing sheep and things like that. And so they used to call it wool sorters disease. They get sores on their hands, but there's a form that's inhaled that's particularly deadly. And he developed something we're going to comment on down the road when we talk about how disease spread called Koch's postulates. And basically, it was like a chain of evidence, like a cause and effect. He basically the animals would die. Okay? And as they would begin to die, they'd have sores all over them. He'd take fluid out of the sores and he'd grow them in the lab on agar plates, like you were doing. And then he would isolate the cultures that came out. he'd inject them into healthy, susceptible animals. And if they died in the same fashion with the same cures, then basically he felt on his cause and effect. And for the longest time, that was the way we identified cause and effect in medicine for infectious disease. We had to conform. We don't use them so much anymore because his premise was you had to be able to grow everything in pure culture. And the reality is you can't. And not all diseases are single organisms. There's a lot of diseases that are multiple organisms. And one of the most famous diseases of them all is still around today, but every kind of waxes and wanes. It goes up and down depending on how cognizant we are of syphilis, which is a bacterial disease, sexually transmitted almost exclusively. Bacterial disease, which you can't grow in the lab. Even with all the modern technology, we still haven't been able to grow it in pure culture. So by that rationale, it can't conform to Koch's postulates. So there's a ton of stuff from all of this. So I thought I would share some of that with you in the history of these microorganisms. So as when we look back at them, Tyndall and Cohn and those guys, so there was quite a dispute, and steriline broths different times. This was the hay infusion nugget that I was telling you about. Cohn, who worked with Koch, he was the one, the heat-resistant forms of bacteria, and there's the difference between sugar and yeast. And so, and really for validation for when you're doing someone else's experiments to either prove or disprove, you have to use the same conditions. And I never go through this golden age of microbiology, but if you're interested, there's a neat timeline that tells you about some of the fascinating things that have occurred with regard to that, which will bring us back to the PowerPoint we're using in Cal. So that's the historical nugget. So, and this is their take on it, their way of expressing it. So, flask with broth shaped the opening into swan necks. His rationale was that the dust that gathered in that little bend was really where the problem is. But he had it exposed to air, which was the whole idea. That was the most persuasive part, and still there were doubters because that's the nature. Okay. From a microscopy standpoint, this is, I know we mentioned that before, Hook and von Leeuwenhoek. If we didn't, I might have told to the other group. Hook was really, he looked at plant and cork and was able to really the first one to draw the cell. He was the original compound microscope, where von Leeuwenhoek had a single lens microscope that a lot of colleges used to make for fun in the micro lab. And basically, he did drawings because he wasn't a scientist; he was a fabric merchant. He used to look at fabric, but in between trips, buying trips, he looked at things in water, and it really did a lot of magnification. And then all of these, the hand washing things, aseptic technique, etc. And so, again, just to summarize, Pasteur much more about finding ways to efficiently treat people, where Koch was much more about, how should I say it, the laboratory aspect, things like that. I mean, he was the one when in the lab, and I usually do this in lab, you use something, when you talk about agar is an extract, it's effectively a seaweed extract. Interestingly, the reason we can use it in the lab is bacteria don't eat it. I mean, years ago, they used to use gelatin. Okay, but a lot of bacteria will have enzymes that'll gobble up gelatin. So since it's not a nutrient, they grow on it, and it demonstrates their characteristics. And it's very amenable to heating and dissolving stuff and then firming up relatively quickly. And interestingly, the way it was developed I used it in the lab, one of his colleagues, the wife of that colleague was a chef, and she made molds. And when they used Jello molds in the heat, they would and when they used agar, the molds would maintain their shape even on warm days. That was the good news. The bad news is agar don't taste good. So if you try, even though it looked good, if you try to eat a slice of it, it was like, whoa. Just say. But I'm a big believer there's always room for Jello. So nobody cares about your humor. Okay. Interesting things. I made a little note about this when I talked about plasmids. Restriction enzymes are only from bacteria. Without them, we would have effectively no genetic modification. We couldn't do DNA testing. We couldn't do them forensically. We couldn't do paternity testing. It's all because of restriction enzymes. Enzymes that nearly all bacteria have, that not only do they chop up DNA, they chop up DNA very specifically. If you don't recall it, real quick, DNA only has 4 letters: A's, T's, G's, and C's, right? And it reliably chops them at places specifically. And what it creates, and remember, DNA strands are what we call anti-parallel. Remember going over that in the laboratory? One is right side up, one's upside down. So they're identical strands. One's right side up, one's upside down. And the letters correspond at certain areas where you get repeat patterns. And it chops them in sort of this zigzag portion. And because we call them, for whatever reason, they call them sticky ends. So you can, in the laboratory, create a section of DNA that will adhere based on these patterns. Okay. And really, when you hear about any of those types of things, DNA testing, whether it's used in a courtroom, whether it's paternity testing, whether you have fun with one of those ancestry.coms or 23andMe's, which are very popular for a lot of folks, right? You can find out about it. So the biological revolution of the 21st century. And it really was the hallmark of something called PCR, which we do here for our advanced students, cell and molecular and biochemistry students. We have PCR machines. If you're wondering what they're doing over in the gym over there, we're making an advanced biology laboratory. So that's, as money is available, we're going to basically be converting aspects of that gym and expanding some of the laboratories. That's the first big project that's going on. So we do this, and basically we can take a teeny tiny amount of DNA, like a teeny tiny amount of blood or some type of fluid, and it's almost like putting it in a photocopier. The enzyme was discovered by a crazy guy who won a Nobel Prize named Dr. Kerry Mullis, and he was just out there and brilliant, but out there. And he found that this enzyme could reliably make vast amounts of identical DNA. And then by utilizing that, he developed this PCR, which stands for polymerase chain reaction. Not that you have to know that. And so it really became significant that was there. And there's other stuff with sequencing, that the reality is that most of our DNA, the vast majority of our DNA, doesn't directly code for proteins. It's not really genetic in nature. 98% of all of our DNA is pretty much the same. There's a small amount that differentiates you and me and anybody else in our physical characteristics or other characteristics. So much of it doesn't. A lot of it's regulatory. They used to erroneously call it junk DNA, but the reality is it's very much, it's regulatory and fragments that other things attach to that kind of trigger the process. So it's not like it's, because if you've ever seen a child with a genetic defect where they're missing a piece of a chromosome, trisomy 21, Down syndrome. Okay. There's a variety, where there's irregularities or loss. It's not good. Okay, so they're critical roles in regulation, etc. So they're that. And this is, we'll get to this a lot in the context of this course. The reality is that we're running out of antibiotics. And the antibiotics that we're using, the bacteria are becoming more and more resistant, developing resistance all the time because we've overused them. And I'll go into this in chapter. We've got to find other ways to treat them. We're actually looking at different things to be able to address that. They're looking at, interestingly, viruses that attack bacteria called phage viruses, which we'll discuss as alternatives. These RNAs, important in bacteria, because they can be targets for antimicrobials, because if they're in the bacteria and not in us, specifically, it could be treatment that doesn't affect us but can destroy the bacteria. And so this is where the science is interesting, because it does things for good and sometimes it backfires. Would be the best way to describe it. So the Human Microbiome Project was a project that was started. I'm trying to see, it was like the DNA sequencing project during the Clinton administration. And so there's a lot of these projects where we're trying to get an idea of what's typically all over us, all the communities, the similarities, how they impact, and studying which ones are present or absent or in combination as they affect our health or our body size or our propensity to get certain diseases. So we're beginning to appreciate the interaction. I mean, and this is one of the large problems that we'll talk about. It's something called the hygiene hypothesis that became more and more apparent, and it's why I get a little preachy on on your generation that was basically shut in for a significant period of time vis-a-vis COVID. I mean, it's so foreign. A little bit more history. Perhaps the most interesting characters in English history were Henry VIII and Elizabeth I. Henry VIII had a bad habit of killing wives. A lot. 60.
That is a bad habit.
Yeah, we didn't kill them all, but he was close. Okay. Anne of Cleves and Catherine Parr survived. But other than that, it wasn't good to be either. Got just stop. Long story. Interesting character. All of which because he wanted a son, because he wanted an heir. So his third wife, Jane Seymour, gave him an heir. And she died of childbirth fever, which interestingly his mom had died of. And so he became the immediate successor to Henry VIII. And he was, because they were so worried about him getting sick, they isolated him from all other kids. Mark Twain wrote a book called Prince and the Pauper, kind of about that. That was very, very interesting. It's done as a movie a couple of times. And they would wipe down the walls with lye and disinfect 3 times a day. Wouldn't let him play with other children. They were deathly afraid if he got a fever, he would die. They didn't have any antibiotics, they didn't have any treatment. As opposed to the people who were kids in our day and age, they threw us out of the house. You get home from school, just go out, go play, come back at dinner time. To be fair, my parents weren't worried about somebody snatching their kids. Every mom that was home, and they were mostly stay-at-home moms, were watching out. And if you did something bad at the playground, half a mile away, by the time you got home, your mom knew about it. God forbid, when your dad got home, you were in trouble. They didn't get that memo about not striking children. They didn't hear that. So, see, when I tried out with my kids, many of you have heard this before, okay? I go, you like this, and you go this, and they go, 1-800 for a child. They do that number. What goes around comes around. They get frisky with me. I go, elder abuse, elder abuse. You heard that before, didn't you? You did too. Rosabella, right? See, you can run, but you can't hide.
You're not elder though.
God bless you. There's a special place in heaven for people who are nice.
My great.
Don't start. I think I like you better than the other group.
Really?
You joking. So, and von Leeuwenhoek built the first. So the idea of the hygiene hypothesis, Edward, whatever number he was, he died before his majority. In other words, when he became king, he wasn't old enough by their rules to become king. His uncle was basically the regent for him, and he died. And then his older stepsister, Queen Mary, and then she didn't live very long, not for that reason, because she had an abdominal tumor. And then Queen Elizabeth I, and it must be a trend with Elizabeth, lived a real long time. So if you get Netflix, watch The Crown. It's a really good show. It's really good, very interesting. They did a great job on that. Four main families of biochemicals. Why? Oh, this is the biochemistry part. Aha! We did all this. This is AP1. You don't have to do it. Okay. So again, in my world, when you have had something like AP1, which you have to have, or General Bio 1 in order to take this course, you should know this stuff. Okay. So that's not stuff that I review. So I'll talk about proteins, we'll talk about nucleotides and genetics, and we'll talk about proteins a lot, and all of those types of things, but you should know the groups that are there, the difference between monomers and polymers. If you don't, you want to shoot me a question, that's fine. But we're certainly not, I'm not going to go over the three monosaccharides and glucose, fructose, and galactose, or the disaccharides, maltose, sucrose, and lactose, any of that. I'm not going to go over those. They're all here. Fats and oils and things like that. But there's relevance to all of these. Like mycolic acid is a wax. That's part of a capsule that effectively is very, very interesting as part of tuberculosis, which makes it sort of a unique molecule in that regard. And very, very hard to destroy and very, very hard to treat. So a lot of the basis of what we talk about here in micro has to come from in the university setting, in any area of education. You build on stuff that you got. So if it's something, go back and look at it. It's okay. Okay, so we're not going to go into the carbohydrates and all that stuff. Want to, not going to do it. We're not going to go into the polysaccharides. The agar, again, is interesting because of its remarkable ability that you can heat it up to boiling, add stuff to it, and when it cools and it holds. That's the beauty of it in the lab. Chitin, you're going to see that's an unusual thing in fungi. Fungi are eukaryotic, just like yeast, but what it makes chitin is the stuff that like the shell of a lobster is made out of, like a bug shell. Okay, it's chitin. They have an exoskeleton. It's very, very hard. Yes, if you get mushrooms that are like, if you get shiitakes in the store, you can't eat the stems. They're very chitinous. They're useful for stock, but they're not great. They don't make good eats. Just where are you all getting ready to go? And we'll talk a lot about peptidoglycan, and we'll talk a lot about lipopolysaccharides, and a lot about glycocalyxes. So we'll look at all that. I don't think you have to do anything. I just want to finish this. That's rehash. We're not going to do the membranes. That's rehash. Things like that. Phospholayers, protein formations, etc. But that's all stuff that you had, so you should have. All right, I'll stop here. Thank you. Goodbye. See you all. Whole week this week. Be sick of me.
Never. It's not possible.